In this laboratory, you will learn about molecular genetics, the study of the structure and function of DNA. You will examine how DNA functions as the genetic material, how it is replicated, and how protein synthesis occurs. Each DNA molecule is composed of two strands of nucleotides. The strands are held together by hydrogen bonds that form between complementary pairs of nitrogenous bases. The two strands of a DNA molecule spiral around each other to form a double helix. A double helix is a geometrical shape that is composed of two strands that coil around the same axis. The pairing of nitrogenous bases holding two DNA strands together is not random. Adenine only base pairs with thymine, A to T, and cytosine only base pairs with guanine, C to G. This complementary base pairing is the key to the genetic code. DNA molecules are very long. In fact, in each human cell, there is about 3 feet of DNA containing 6 billion base pairs. In the entire body, there's enough DNA to stretch from the Earth to the Sun and back again more than 300 times. As you can imagine, in order to fit this much DNA into a cell, it must be very tightly packed. To package DNA into such a small volume, it is wrapped around histone proteins, forming a highly compact structure called chromatin. The top image shows an electron microscope picture of a DNA molecule. It is 30 nanometers in width. The illustration below shows the interaction between DNA and histone proteins in this structure. As you can see, the DNA, which is colored red on the diagram, is wrapped around histone proteins, which are shown in pink. During DNA replication, each DNA molecule is duplicated to produce two identical DNA molecules. To begin the process, a DNA molecule unzips so that the nitrogenous bases on each strand are exposed. New complementary DNA nucleotides line up along each of the parent strands. The enzyme DNA polymerase links the nucleotides together and then proofreads the strands to be sure they are correct. The result is two DNA molecules, each composed of one parent strand of nucleotides from the original DNA molecule and one new strand. This is why DNA replication is described as semi-conservative.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated, and the DNA replication machinery is assembled at the replication fork. The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semi-conservative DNA replication because half of the DNA molecule is conserved from the original molecule.
the instructions for making proteins are contained in the order of nucleotides in a DNA molecule. An individual nucleotide is shown on the right in the diagram. It is composed of a sugar, a phosphate, and a nitrogenous base. A group of 3 nucleotides in a DNA strand encodes the information for a specific amino acid. Because DNA contains the instructions for making proteins, a complete copy of a cell's DNA must be passed on to each new generation of cells. When a cell is about to divide, it first replicates its DNA. The replication process produces 2 identical copies of each DNA molecule within a cell. Each daughter cell receives a complete copy of the DNA from the parent cell, assuring that it has the genetic information necessary to produce all of the proteins necessary for life. Genetic information in cells flows from DNA to RNA to protein. This path was previously known as the central dogma of biology. DNA contains the instructions, and the 3 forms of RNA take the information and construct proteins through the process of translation, or protein synthesis. RNA differs from DNA in 3 ways. First, RNA is a single-stranded molecule, while DNA is double-stranded. Second, the RNA backbone contains ribose instead of deoxyribose as in DNA. Third, RNA uses the nucleotide uracil instead of thymine. This means that an RNA nucleotide containing uracil will pair with a DNA nucleotide containing adenine, AU pairing. DNA and RNA are structurally similar but serve different functions in cells. DNA functions as the genetic material containing the instructions for making all of a cell's proteins and is passed from parent to offspring. There are many functional types of RNA. We will discuss 3 types that are used in the process of translation. Messenger RNA carries the genetic information, or message, for making a protein from DNA to the ribosome. Ribosomal RNA composes the ribosomes, which are the site of assembly of amino acids into proteins. Transfer RNA transfers a specific amino acid to a growing polypeptide chain at the ribosome. Each chromosome carries the instructions for making many proteins. A segment of DNA that codes for a single protein is called a gene. When a cell needs to make a specific protein, the gene for that protein is said to be turned on or expressed. The first step of gene expression is to make a copy of the gene from the mRNA. This process is called transcription. In reality, many mRNA copies or transcripts are made from a particular gene when its expression is turned on. The next step of gene expression is to convert the sequence of nucleotides in the mRNA, encoded as triplets of bases called codons, into a sequence of amino acids. This process is called translation and occurs on a ribosome.